For our research article, our topic was Hebbian learning and the brain. This project is by Lindsay Johns, Julia Gallagher, and Abby O'Brien. The article we chose to do our research on was Integrating Hebbian and Homeostatic Plasticity by Kevin Fox and Michael Stryker. We were unsure with the term Hebbian learning before this project, so we figured we would explain it to you guys before starting our presentation. Hebbian learning is when our brains learn something new, they form a neural network that gets easier and easier over time. An example of this would be when you are taught to tie your shoe. It is very difficult at first, but as time goes on, it becomes very natural, and you normally don't even think about it when you do it.
The theoretical framework for Hebbian learning is that it's a mechanism by which information is coded and retained within neurons. Hebbian plasticity also plays an important role in fundamental properties of the brain, such as learning and memory, as well as development and recovery from loss of function. When we learn something new, our neurons are activated and they connect with other neurons, creating a network. In this study's research, some questions that were imposed was, how does homeostatic plasticity avoid erasing the Hebbian coded information, as well as what is Hebbian and homeostatic plasticity and why might it be important to integrate them? Our hypothesis was Hebbian plasticity is the mechanism that codes the neurons and retains them, whereas homeostatic is when the neuron moves back towards its original state following a perturbation, which means disrupted routine.
Participants in the study were given 3 papers that discuss the topics of Hebbian learning and plasticity. The creators of the study then identified 4 different types of homeoplasticity. For parts of this procedure, animal models filled in to test these different aspects. This proved difficult at times because the animal subjects did not necessarily have the exact same forms of homeoplasticity as humans. The experiment came to the conclusion that there needed to be experiments designed that would allow probing of synaptic scaling to be able to study these receptors in synapses on a cellular level. The authors looked into the relationship between TNFA activity and neurodevelopmental disorders such as autism or schizophrenia using an MIA model or maternal immune activation model. They found through using model mice that TNFA alpha was not necessary for abnormal social responses. The study also put a strong spotlight on the part of the brain that is distinguishable from other cytokines. The hypothesis was both supported and disputed at times. Some questions were put away and some were brought forward throughout the experiment.
So what do the authors' findings mean? Like we said, the hypothesis was both supported and disputed at times. Some questions were put away and some were brought forward throughout the experiment. Some real-world implications are, the first example we thought of is when you first learn to drive your car, you take notes to make sure you are going the speed limit, stopping for a certain amount of time at a stop sign, turning on your blinker, and doing all the little things. As time goes on, you do all these things naturally without even thinking about it. Another example is when you are a baby and first learn to walk, you learn step by step how to do it. After you learn, it comes naturally to you and you never think about how you need to balance and step 1 foot at a time, you just do it. Another example is the one that I mentioned in the beginning, as when you learn to tie your shoe. At first, it's very difficult, but then it becomes natural to you and you don't even think about it. The question we came up with is, how can teachers make sure they understand how to interpret Hebbian learning in their lessons since it has been shown to be so effective? And here is our citation for our article that we used.